My name is Rohin Iyer, and as mentioned, I am a development manager with GE Healthcare, and I'll be presenting on a closed CAR-T manufacturing process. Just a little bit about the team that I'm a part of. This is a unique collaboration between GE Healthcare and the Center for Commercialization of Regenerative Medicine based in Toronto, Canada. So I am a Canadian, so I will be polite if you come up and ask me questions afterwards, and I'm happy to discuss potential collaborations and opportunities. But what's really unique about this is that this is actually a public-private partnership. We have a team of about 105 people, of which about 36 of us are technical people, scientists and engineers and immunologists who work together in one location, and it's a mix of GE Healthcare and CCRM employees. And CCRM is a nonprofit, so this is quite a unique model, and it allowed us to kind of be very agile and kind of move quickly in terms of our ability to develop a closed CAR-T process. So I wanted to talk a little bit about that. And so the team that we've built here is part of a facility which we call the Center for Advanced Therapeutic Cell Technologies, or the CADCT. And there's Michael May presenting, some of our capabilities. We can do anything from process development and optimization to regulatory to custom media development. And though we do have abilities in terms of viral manufacturing, viral process development, and CAR-T, I'll be focusing more on CAR-T today because that's really the focus of this particular presentation. In addition to our process development facility, we also recently christened our CCVP, or Center for Cell and Vector Production, which is our GMP suite based in Toronto as well. 20,000 square feet of when spread across 10 clean rooms, 2 of which are for viral vector manufacturing. And so we've now newly built up this capacity to address the needs of the cell and gene therapy industry. So why automate? Why do we need to automate CAR-T? Well, it's because if you look at the top left over here, this is kind of how CAR-T process development or CAR-T manufacturing is done in a lot of academic and kind of clinical capacities. But what we really want to do is move to something that's a little more akin to what is used in the bioprocess industry, where there's automation, there's quality control, there's a little more rigor around how a process is run and how it's developed. And so that's the vision, and so many of you know this already, so I don't need to tell you a lot about CAR-T therapy, but if we talk about some of the approved products out there, some of the key challenges associated with them are obviously cost. Some of these therapies run into the many hundreds of thousands of US dollars, and then when you convert that to Canadian, that's even worse, right? So we really want to bring the cost down. We want to, de-risk the process, increase our ability to, bring safety to the patient. And, another key issue is that the length of these CAR-T processes are typically on the order of, 22 to plus days. And what we're trying to do is reduce the time that it takes to actually run that process. So the goal of this project and of this vision that we developed around CAR-T was to kind of systematically and robustly develop an automated or automated as far as it could be process, using commercially available reagents that are GMP available, integrating automation, and also maintaining flexibility across that process. So how we did this was, initially we did small-scale process development, so on a unit operation by unit operation basis. We kind of looked at some of the key initial steps, such as transduction, that initial isolation of the PBMC population, how many days post-activation do you do transduction and things like that. And once we developed all those unit ops at the small scale, then we moved on to test them and validate them at the large scale. All right, so some initial data here was around a comparison of different enrichment methods. So we compared the EasyCEP CD3 positive selection method to the other methods that are out there, so the QuadraMax CD3 positive, as well as the negative selection method using the QuadraMax. So we removed the CD14 and CD19 populations from the PBMC mix, and both of these methods were pretty much comparable in terms of overall recovery, purity, and viability. But what we noticed was that when we did transduction, either day 1 or day 2 after activation had been had happened, we noticed that there was a significant difference in terms of the ability to transduce these cells with a GFP encoding LVV. So we got higher transduction efficiencies in, let's say, the CD3 positive selection method versus the CD14 and CD19 depletion method. And there was also no statistical difference between the two different kits that we used to do this in the small scale. And then looking a little bit deeper at the different transduction methods, if we compared, let's say, what's typically done in the field, which is either using static cultures such as a T-flask or a retronectin-coated well plate, versus a more agitated or a more active method of transduction such as a shake flask or a spinoculation method, we noticed that there was really no statistical difference between these groups, but there was certainly a trend towards spinoculation being having slightly higher transduction efficiencies, even over 70% in many cases. Again, this is a GFP encoding LVV, but proof of concept is that if you did this with a CAR encoding LVV, you would achieve similar transduction efficiencies. And similarly, in terms of the viability, relative fold expansion, and difference between CD3, CD4, and CD8 populations, we didn't notice any differences between the shake flask versus the T-flask. So this motivated us to kind of move away from using the kind of open and manual T-flask method and move toward a more active and closed shake flask method to do all of our subsequent activation and transduction. And so now we integrated all of that knowledge that we had developed in the small scale into a large scale process where we integrated a lot of the GE equipment into the process. So we used the SmartMax for that initial wash of the apheresis unit, which was a frozen apheresis unit in this particular process. CPAX-2, which we now have the equivalent CPAX-C Pro for manufacturing. And then moving from PBMC into EasySEP CD3 positive isolation, activation, as I mentioned, in the shake flask, and then transduction in the CPAX-C Pro, which is a closed transduction method, expansion of the Jury rocking bioreactor for several days, and then downstream processing of the Cephia, which is our downstream processing unit that does volume reduction, washing, and a buffer exchange. And then we've also got some products in the cryopreservation kind of part of the downstream, which include the ViaFreeze, which is a liquid nitrogen-free cryopreservation system, as well as the ViaThaw, which is a dry thawing system. So comparing different methods across multiple biological replicates, we saw absolutely no real difference in terms of PBMC recovery and viability compared to the manual methods that we were initially testing, and we got very high CD3 recovery, purity, and viability across different biological replicates. So this was a promising outcome. And then when we took all of those small-scale cultures and put them into the Jury bioreactor, we got at least a minimum of 1e⁻⁻⁻, or essentially 10 billion CAR T cells at the end of the process. Obviously, there is some biological variability as you go from donor to donor. These are all healthy donors as well, so you can expect there to be even more variability perhaps with the patient population. But suffice it to say that we maintained a high transduction efficiency with the GFP encoding LVV with high viability, and we're meeting a minimum dose of 1e⁻⁻⁻, which is far, far above what most people are using in terms of autologous CAR T therapy right now. And then just looking at the downstream, we also put those cells into the Cephia, which, as I mentioned, does volume reduction, washing, buffer exchange, and also cryoformulation. And we got a very, very high viability, both pre and post Cephia, so there was no loss of viability coming out of the Cephia, as well as a reasonable recovery of 95%. Recovery post-thaw was a little bit impacted just because we expect this as you cryopreserve cells. There is going to be a little bit of a hit, as you recover them from thaw. But that did not impact their GFP transduction efficiency. So, these cells were viable. They were very much expressing their construct, their transgene, and this was replicated across multiple biological replicates. And so in terms of addressing future demand, it's important to talk about how this is going to impact, let's say when you're in a phase 1 trial versus in a phase 2 or phase 3. There are other companies out there who shall remain nameless and who are our competitors who use this sort of one-box approach where you put everything into one box and you spit out all your CAR T cells on the other end. That works great if you're treating one patient at a time, but what happens is you start to treat more patients and you move into your phase 2 clinical trials is that you start to run into huge bottlenecks unless you buy multiples of these one boxes. Whereas if you use a little more of a scheduled approach, a more of a staggered approach, half of these unit operations are being used for maybe 1 to 2 hours at a time, and then they can be freed up to do more work for other patient populations. So what we really would like to see is to be able to treat more patients in a more efficient manner and to be able to schedule it in such a way that you don't really need all your unit ops tied up all the time. And that's kind of the approach that we take with the GE systems for CAR T therapy. So I think I've ended a little bit early, but I made up for the overtime on the previous presentation, but maybe we can use the extra time to take some more questions because I think it's more about the conversation, and I'm happy to take questions at this point, so thank you.